My work deals with one of the main problems for the production of white and sparkling wines under the climate change scenario. In the specific, the fact that in the most hot season, we commonly find very low acidity in grapes and in wines due to the high respiration rates affecting the malic acid concentration. We find very low malic acid concentration. In this context, many growers or many districts are today reconsidering if grow different varieties as compared to the most commonly grown to improve the acidity in grapes at harvest. In this work, we have evaluated over 4 seasons the yield, productive parameters, ripening parameters of 2 different cultivars, Ortrugo and Barbesino. And we determined the seasonal dynamics of many ripening parameters with the goal of understanding which parameters could better determine the genotype attitude in maintaining an adequate acidity at harvest. The general goal is to find new benchmark for choosing cultivars at farm level, at district level, or maybe for the process of breeding or phenotyping cultivars directly for the acidity, these issues. Over 4 seasons, the 2 varieties, Ortrugo in blue and Barbesino in red, had a very similar sugar accumulation pattern. However, Barbesino, the cultivar in red, steadily maintained higher acidity from preparation until the end of ripening in any of the 4 seasons. In the bottom corner on the right, you can see the plotting of sugars on acidity, and Barbesino maintaining higher acidity for any sugar level. Well, this higher acidity was mainly due to malic acid concentration. Malic acid was higher in Barbesino with a very similar pattern as compared to titratable acidity. Well, when we analyzed malic acid degradation rates, we were expecting a little bit the opposite. Essentially, Barbesino was the varieties exhibiting the higher maximum malic acid concentration rates in almost all the years. And if considering the period after the peaks, Barbesino is the one that was having higher malic acid degradation rates in grams per liter per day consistently up to the end of, until the end of the season in any of the 4 years evaluated. When we plotted the malic acid degradation rates, so the gram per liter per loss per day in terms of malic acid with the instantaneous malic acid concentration, so essentially analyzing the loss of malic acid in terms of the instantaneous concentration of malic acid, a very steady and consistent correlation came from this analysis. In short, even if Barbesino was having a very higher malic acid degradation rates for values between a very high level of malic acid to values of around 10, considering the different years, this trend was essentially reversed when the cultivars were approaching the final part of the ripening. So when the malic acid concentration was lower. This was very consistent in any of the 4 different seasons. Extrapolating for any of the replicates for any of the year, the 2 different parameters, so the malic acid degradation rates at a malic acid concentration of 5 grams per liter and the malic acid degradation rates when malic acid was 2.5 grams per liter, we had these results with Barbesino having steadily lower malic acid degradation rates. In conclusion, our work suggests that sugar and variation time cannot really be considered a good parameters to analyze final acidity at harvest, because sugars and acidity can have a very distinct and different pattern during ripening. Malic acid degradation rates can also be maybe a misleading parameters if not analyzed correctly, because can happen that the varieties maintaining higher acidity at harvest due to a higher level of malic acid preparation exhibit higher maximum concentration rates right at variation and later. What our work demonstrates that there is a strict genetic control over malic acid degradation rates at varying malic acid concentration level. This relationship is very consistent over different seasons with different weather pattern. Our 4 seasons were very different each other. So in our opinion, this parameter, the analysis of malic acid degradation rates in terms of their variation over the instantaneous malic acid concentration can be a good parameter in order to be introduced when selecting new cultivar or introducing new cultivar in different environments, or maybe for finding, maybe in the process of breeding new cultivars when selecting accession in the progeny, for instance. Thank you very much.